Hello everyone, myself Professor Shahana Sengupta. Presently, I am associated with Swami Vivekananda Institute of Modern Science, Kolkata as an assistant professor in the Department of Microbiology. I want to welcome you all in my YouTube channel where I used to share different academic videos related to different field of biological science. I also want to convey my heartfelt thanks to all my viewers and my subscribers. Today, my topic of lecture is acquired immune system. So let's start our journey with the new topic. Happy watching. Let's discuss about the acquired immunity. Acquired immunity also can be called as adaptive immunity or specific immunity. As this particular type of immunity, they are very much specific against a sort of antigens or foreign immunogens. That's why they are known as specific immunity. Adaptive or specific immunity is quite capable of recognizing and selectively eliminating specific foreign antigens and related molecules. Let's talk about the components of acquired immunity. Acquired immunity comprises of two major ingredients. One is lymphocytes, another is antibodies. Lymphocytes are categorized into two major types. Number one, B lymphocytes or B cell, and number two, T lymphocytes or T cell. Both the B cells and T cells, they are being originated from the bone marrow, and after that they are being developed in the bone marrow and thymus respectively. Antibodies are being produced from the plasma kind of B lymphocytes, and after being production they used to present in the physiological fluid of the host body system. Now I would like to discuss about the characteristics features of acquired immune response. Acquired immune response comprises of four major characteristic features. They are namely specificity, adaptiveness, capacity of making discrimination between the self and non-self molecule, and lastly the capacity of memorization. First, we would like to start from specificity. Specificity is the ability to discriminate among different molecular entities and to respond only those uniquely required. Number two is the adaptiveness. It is the ability to respond to previously unseen molecules. Number three, discrimination between self and non-self. It is the ability to recognize and respond to molecules that are foreign or non-self and to avoid making responses to those molecules they are self. This discrimination is conferred by specialized cells that are known as lymphocytes that bear on their surface a specific antigen receptor. The fourth one is memorizing capacity, the ability to recall previous contact with a foreign molecule and to give response accordingly in a more learned manner. That means with a more rapid and larger response is the functions of the memory cell. Memory cells are the boon of the immune system, and they memorize the whole phenomenon that is going to happen within a immune system after our immune system basically facing the challenges coming from the antigen or any foreign entities. Now I would like to discuss about the types of acquired immunity. Acquired immunity divided into two major types. Number one, naturally acquired immunity, and number two, artificially acquired immunity. The naturally acquired immunity again subdivided into two major types. Number one, naturally active and naturally passive acquired immunity. Whereas the artificially active immunity, they are also subdivided into two major types. They are of similar types. Number one, it is the artificially active acquired immunity, and number two, that is the artificially passive acquired immunity. Now I would like to discuss about the each and every types of the acquired immune system in a quite detailed manner. Firstly, I would like to start with the naturally acquired active immunity. It occurs when the person is exposed to a live pathogen, develops disease, and becomes immune as a result of the primary immune response. Once a microbe penetrates the host body's skin through mucous membrane or other primary defenses, it interacts with the immune system. B cell that is present in the host body and responsible for the productions of antibodies that help to fight against invading microorganisms. The adaptive immune response generated against the pathogen takes days or even weeks to develop but may be lifelong. Wild infection, for example, with hepatitis A virus like HAV and subsequent recovery gives rise to a natural active immune response, usually leading to a lifelong protection. The next important one is artificially acquired active immunity. It is the protection that is produced by intentional exposure of a person to a antigen in a vaccine mode so as to produce an active and lasting immune response. During the time of the vaccination, there occur the utilizations of attenuated form of the particular causative agent. The third one is naturally acquired passive immunity that occurs during the pregnancy, in which certain antibodies are passed from the maternal blood into the fetal bloodstream, mostly in the form of IgG. Antibodies are transformed from one person to another through natural means such as in prenatal and postnatal relationship between mother and child. Artificially acquired passive immunity, that is the fourth one. This is an immediate but short-term immunization provided by the injection of crude antibodies such as gamma globulin. They are not produced by the recipient's body itself. These antibodies are developed in another individual or in animal body and then injected into another individual for the cure purpose or the treatment purpose. It is generally called as antiserum. The most widely popular example of this artificially acquired passive immunity, it is the antivenom injection that is given to the host body just after snake biting. Now I would like to discuss about the general mechanism of acquired immune responses. The general mechanism of acquired immune responses can be described via few steps. The adaptive or the acquired immune response is a very specific one, induced by a given antigen, and that generates a response that specifically targets that same antigen. It takes place after the failure of the innate one and responds specifically starting about 96 to 120 hours after the infection. Let's start with the first step under this mechanism. It is the recognition of the antigen by specific lymphocytes. Lymphocytes are very unique components of the acquired immune responses. Two different types of lymphocytes, namely T cell and B cell, they are the main two ingredients of this type of responses. Both are being originated from the bone marrow, get matured in thymus and bone marrow respectively. Both two types of lymphocytes, they are equipped with two cell surface receptor. In case of T cell, it is known as TCR, and in case of B cell, it is known as BCR. These receptors are really playing a very important role in first recognition of this antigen. They are very much unique and they are having specific affinity towards the antigen. That's why they can easily recognize the antigen with their specificity. The antigen, whenever it enters or invades our host defense system, they are being presented with the help of APCs or antigen presenting cells. Along with antigen presenting cells, they are also being expressed with the help of another sets of protein molecules, namely MHC. Depending on the types of the antigen, there will be the attachment of either the MHC class 1 molecule or MHC class 2 molecule with the antigen. The MHCs are nothing but a group of protein known as major histocompatibility complexes. After being recognized, there occur the activations of the both lymphocytes. Whenever the lymphocytes get activated and after their proper activation, they are ready to interact with the antigen by the productions of antibodies from the B cell and chemokines and cytokines in the form of interferons and interleukins from the T cell. In the third step, after activation, the lymphocytes they undergo proliferation. In these steps, there occur the increase in the number of both these two types of cell. And after the proliferation step, there occur the step of differentiation. In these steps, both the lymphocytes they get differentiated into two different cell types. The B cell get differentiated into plasma B cell and memory B cell, whereas the T cell get also differentiated into the effector T cell and the memory T cell. The plasma B cell responsible for the production of antibody and the memory B cell is responsible for memorizing the whole phenomenon or the whole situation that is taking place during this particular kind of responses. Whereas the effector T cells, they are responsible for the productions of chemokines and cytokines, whereas the memory T cells basically functioning just like the memory B cell. That means the main function of the memory T cell is to memorize the whole phenomenon or the whole reaction that is taking place during the initiation of the immune responses. Now I would like to continue with the general mechanism of acquired immune responses. In the fifth point of this mechanism, I would like to discuss about the elimination of the antigen. When antigen infects the host defense mechanism and face the reaction of different types of lymphocytes, at the last of the reaction, antigen has to be eliminated from the body of the host as a result of the immune initiation. And those antigens or the infectious agents, they have not been eliminated during the innate response. They are subjected to reaction under this acquired one. After the elimination of the antigen, there occur returning of the homeostasis in the host body, as well as there also occur the development of the memory cell as B cells and T cells are being produced as the part of the memory B cell and the memory T cell. Memory cells are basically regarded as the boon of the immune system, and memory cells can evoke a more rapid and long responses on re-exposure of a same antigen. Now I would like to discuss about the immunological memory, which results from the antigen specific B lymphocyte and T lymphocyte as the production of memory B cell and memory T cell. It is basically very important characteristics of adaptive immunity. It can be called as a cardinal feature of this particular immunity. It means that the immune system can remember the antigen that previously activated and launch a more intense response reaction when encountering the same antigen for a second time. The memory cells have a long lifespan and remain in our body for a considerable long period of time. That's why it is very important in the vaccinological experiment. The goal of vaccinology is to introduce long-lived immunological memory component within the host body, and that can give protects against reinfection. Now, I would like to continue my lecture with the discussion related to humoral immune response, or in abbreviation HIR. In general, humoral response refers to the responses which is mediated by molecules. That means that it is possible to transfer the particular kind of immunity from one organism to another just by transferring the responsible molecules. And these molecules are mostly antibodies or the serum antibodies that is produced by the plasma B cell. More specifically, someone who has never been exposed to a specific disease can gain humoral immunity through administration of antibodies from someone who has been already exposed and survived from the same disease. Basically, the humoral refers to the bodily fluids where these free floating serum antibodies bind to antigen and assist with the elimination. After the reaction, at the end, there occur the destruction and elimination of that extracellular microbes or their toxins in form of the antigen. As well as similarly, there occur the development of memory cell with the help of memory B cell. Let's explain the illustration of humoral immune response. Here we can see an antigen is trying to invade the host defense barrier. By doing this, antigen first is facing the interaction with the lymphocyte. And here we can see the B lymphocyte is facing the antigen, so that the B lymphocyte during that recognition process is already equipped with the specific receptor system, that is the BCR, and along with the particular MHC class 1 or class 2 that is very much helpful in presenting the antigen in front of the B cell. So antigen before presentation, it is already equipped with the APCs and the MHCs, either the class 1 or the class 2, depending on its types. Whenever antigen get presented in front of the B cell, it becomes very easy for the B cell to make interaction with the antigen with the help of its receptor system. Whenever the particular B cell get activated and started taking action against the antigen, similarly there occur the activation of the T helper cell. Basically, T helper cell try to promote the antibody production within the plasma B cell. Whenever the plasma B cell, after recognition and activation, has started production of the antibodies, then the antibodies easily can cause the further damage of the antigen, so that antigen will be eliminated from the host body and homeostasis of the host body will be retaining. Now I would like to continue with cell-mediated immune response, or CMIR. CMIR can be acquired through T cells from someone who is immune to the target disease or infection. Cell-mediated refers to the fact that the response is carried out by the cytotoxic T cell. Much like humoral immunity, someone who has not been exposed to a specific disease can gain cell-mediated immunity through the administration of Th and Tc cells from someone that has been exposed, survived the same disease. So it is clear to us that T cells or the T lymphocytes, they are playing really a very key important role in administration of CMRI. They are basically engaging in recognizing the peptide antigen with the help of TCR, that means T cell receptor, and the surface marker CD or cluster of differentiation. Macrophages and another APCs are also helping in the process of cell-mediated immune response. Along with the APCs, another group of protein molecules, those are known as major histocompatibility complex, either the class 1 or the class 2, are also playing a very important role in cell-mediated immune responses. Now I will continue with the explanation of the illustration of cell-mediated immune responses or CMI. In case of CMI, as we all know that T cell is playing the key role. Whenever antigen is entering in the body of the host by invading the host defense mechanism, that is first being recognized by different types of APCs or the antigen presenting cells like dendritic cells and macrophages. Along with the APCs, another group of protein molecule like MHCs, they are also playing a very important role in recognition and processing of the antigen. For this recognition process, T cell get help from the TCR or T cell receptor and a particular kind of surface marker known as CD or cluster of differentiation. After recognition of the particular antigen, APCs present the antigen in front of the T cell, specifically a subtype of T cell that is known as T helper. As a result, the T helper cell or T8 cell, they interacts with the processed antigen and get activated. That activated T8 cell may cause further activation of two types of cell. One is the B lymphocytes or B cell, another is the cytotoxic T cell or TC cell in abbreviation. The TC cell basically acts on the target, that means acts on the antigen, and thereby cause the further elimination of the antigen from the body of the host, or it may cause further damage to the antigen. Whereas the T helper cell, whenever activates the B cell, there occur particularly the activation of plasma B cell, so that plasma B cell started production of antibodies. So antibodies again help the cytotoxic T cell for the further degradation of the antigen. Thanks a lot for your patience. Please do subscribe my channel. Stay tuned for the upcoming videos in different fields of biosciences.